So here we'll look at how DNA is packaged into chromatin and how different chromatin structures are found in different regions of the cell's genome and how this affects gene expression. And so the linear length of the human genome is about 2 meters. And so it needs to fit into a space that's just 6 microns in diameter. So that would be like trying to fit a 60-mile-long spaghetti noodle onto a dinner plate. So besides the fact that it just won't fit, a really long linear DNA molecule is likely to break and become tangled unless it's organized. And so eukaryotic cells solve these problems by packaging DNA into a compact structure known as chromatin. And the basic unit of eukaryotic chromatin is the nucleosomes. So each of these beads is an individual nucleosome, and then multiple nucleosomes are sometimes represented as beads on a string. If we zoom in, each individual nucleosome or bead is made up of an octamer that contains 2 copies each of 4 histone proteins. So 2 each of H2A, 2 each of H2B, 2H3, and 2H4. And so all of these histones are compact globular proteins. And although you can't see it here, they have a little tail that's rich in positive charge. And so the positive charge on the histone interacts with the negative charge on the DNA phosphate backbone. And each nucleosome core particle or bead, which is the octamer plus the DNA, is around 10 nanometers wide. Adjacent nucleosomes are often packaged into higher order fibers, and these fibers are further organized into loops, and the bases of these loops are attached to protein scaffolds. And these loops are packed together into a regular, tightly condensed structure immediately prior to cell division, like this condensed section of a chromosome. And you can see that how that section relates to this entire metaphase chromosome that has maximum DNA packing. All of this higher order organization and spacing and packing of adjacent nucleosomes varies from gene to gene and also varies over the life of the cell. So let's look at that now. So the organization of DNA into nucleosomes and chromatin makes it more compact so it can fit into the nucleus, but it also raises another issue. So very compact DNA is less accessible, and so the proteins that are involved in replication and transcription aren't able to interact with the DNA. So this compaction and condensing is a barrier to replication and to transcription. And so regions of the chromosome that aren't transcriptionally active largely retain this highly condensed structure, and this is known as heterochromatin. So heterochromatin is considered silent. It often includes repetitive sequences like centromeres or telomeres, for example, as well as regions containing genes that aren't necessary for that particular cell type. So tight packaging of DNA into heterochromatin generally silences the genes within it. But after cell division, the cell needs to loosen the chromatin a little in some areas to allow proteins to come in so that gene transcription can happen. And areas where the chromosomes are more loosely organized and less condensed and have actively transcribed genes are known as euchromatin. And so this process of loosening the chromosome also creates an opportunity for regulation, because the amount of a gene that's transcribed can be altered by making the gene more or less accessible to that transcription machinery. So cells dynamically regulate this chromatin state condensing chromosomes for easier segregation during cell division, then loosening and stretching out certain parts of the chromosome after cell division to allow for localized transcription of genes required for that particular cell type. And a big part of the regulation of chromatin state happens at the level of the nucleosome. So by altering how tightly the octamer associates with the DNA, as well as how tightly adjacent nucleosomes associate with each other. And so the ability to adjust nucleosome positioning in chromatin is called remodeling. And since nucleosomes are typically pretty stable, adjusting their position requires some work. So there's a multi-protein complex, and those complexes typically use energy from ATP hydrolysis to perform remodeling. And so remodeling complexes are also important in the assembly of new nucleosomes on recently replicated DNA as well. So as we've seen in the chromosomes of eukaryotes, DNA is uniformly assembled into nucleosomes, but a variety of different chromatin structures is possible. And this variety is based on a large set of reversible covalent modifications of those four histones in the nucleosome core. And so these modifications include acetylation, methylation, as well as phosphorylation. And the remodeling complexes often have highly specific histone modifying enzymes. So these include histone acetyltransferases, or HATs, that add acetyl groups, and histone deacetylase complexes, or HDACs, that remove them. A set of different histone methyltransferases can add methyl groups, and histone kinases can phosphorylate the histones. And so each enzyme is recruited to a specific site on the chromatin at defined times in each cell's life. And so a large number of these modifications happen on the relatively unstructured and accessible N-terminal histone tails that protrude from the nucleosome, and they can directly mediate the interaction between the histone and the DNA. So both acetylation and methylation of histones are important for the assembly of new nucleosomes after DNA replication, as well as for regulating transcription. So acetylation on the N-terminal tails loosens chromosome structure, partly because adding an acetyl group removes the positive charge, which reduces the affinity of the tails for the adjacent nucleosomes. So histone acetylation transforms condensed chromatin into a more relaxed structure that allows space for transcription factor binding and is associated with greater levels of gene transcription. And then this relaxation can be reversed by HDAC activity, which removes those acetyl groups. Methylation, on the other hand, generally causes the nucleosomes to pack more tightly together, and as a consequence, the transcription factors can't bind the DNA, so the genes aren't expressed. And it does this in part by affecting recruitment of other proteins to interact with the chromatin, but depending on the exact location where the methyl group is added and the number of methyl groups that are added, it's also possible for methylation to activate transcription. And then finally, there's histone phosphorylation, which is known to be involved in DNA damage repair and is important for chromosome condensation just before cell division. So the bottom line is that very different patterns of covalent modifications can be found on different groups of nucleosomes. And so depending on both their position in the genome and on the history of the cell and these histone modifications, they're carefully controlled and they can have important consequences for gene expression.